All of us are scared of cancer, and we have a good reason to fear it. That is because the chance of having a form of this disease in our lifetime are about 50%. That means half of us in this room will get that dreaded diagnosis. And despite fantastic breakthroughs in cancer treatment, it is predicted that more than 600,000 people, which is 3 times the population of Augusta, will die from cancer this year in the United States alone, and millions worldwide. Cancer starts when a group of cells in our body grows and divide very fast and without control. And when this happens, they form solid tumors. These cells can happen in the breast, in the lungs, and even in our brains. When cancer cells divide, they need to make new DNA made up of a lot of nucleotides, so that each new cell will have an identical copy of DNA. This cancer treatment affects the DNA by breaking this DNA into tiny pieces, and the DNA is the support of our cells. This treatment attacks cells that are dividing quickly, shrinks the tumor, but most times the cancer comes back. That is because not all the cells in the tumor are rapidly dividing. When the cells grow away from the blood vessels, it results in areas that have low oxygen, which we refer to as hypoxia. Those areas are slowly dividing and slowly growing. It is also known that low oxygen causes the cancer cells to lose their nucleotide, which, if you recall, is needed to make DNA. So, my research is trying to make those sluggish hypoxic cancer cells to divide more quickly so that they too can become vulnerable to DNA damaging agent. So what I did was that I put some cancer cells in a hypoxic chamber, which is a small box with low oxygen level, just like in the blue region, and I found that the cells stopped dividing. Then I added nucleotide, and I found that my idea was right. The cells started dividing quickly, even without oxygen. The next thing I did was, after causing the cells to start dividing by adding nucleotide to the hypoxic cancer cells, I exposed them to common cancer treatment, like radiation. And I noticed that there was more DNA damage, more cell death in this group of cells compared to the hypoxic cancer cells that had low nucleotide. We are so excited with this result, and we are looking for ways in which we can increase the nucleotide level in mouse tumors. If we are successful at treating all the cells in cancer by pushing even the sluggish hypoxic cancer cells down the cliff of death, then we'll see a day in the near future where we no longer have to fear cancer. Thank you.